This is a project that we worked on, me as a member of Gadi Getz's group at the Broad Institute, but with Marcella Maus at MGH and Kathy Wu, Dana Farber, looking at patients treated with these two CAR T products. And as you may know, CAR T therapy has shown a lot of success in lymphoma. So about half of patients that are otherwise untreatable with standard lines of therapy have these long durable responses. But there's still these half of patients that aren't seeing this maintaining response after the course of treatment. So what we really wanted to do is to hone in on the T cells themselves, as there's some preliminary data in other tumor types and on certain products that the T cells themselves, rather than the tumor, may hold a lot of the information about why patients don't respond. And so we used single-cell RNA sequencing technology to really follow the T cell before treatment to after treatment, as well as looking at the infusion products themselves, so the actual CAR T product that these patients are treated with. And by doing this, we're able to show that these two products have very different dynamics in terms of how they look in responders versus non-responders between the two different products. So when we look at tisa-cel, we see these large changes in dynamics between at baseline versus the infusion product cells versus post-treatment, and contractions and expansions of these cytotoxic CD8 T cells that have a specific phenotype in the infusion product. And we find that this expansion is very predictive of response for tisa-cel, but we don't see the same thing for axi-cel. For axi-cel, we more or less see the dynamics within a patient tend to be much more stable between what we see in the infusion product and what we get post-treatment. In the axi-cel cases, we found different populations to be important. We are particularly interested in these CAR regulatory T cells. So these are T cells whose function is to suppress immune responses and normally to prevent autoimmunity. But in this case, we think that these cells could be holding back patients treated with axi-cel from being able to exhibit a full response because this suppressive force is there. And another interesting point that we were able to look at is how CAR T cells sort of work across multiple treatments. So it's fairly rare for a patient to get a second dose of CAR T therapy. But in one of the cases, it happened in our cohort, and we were actually able to do single-cell sequencing both before the first dose and or after first dose and after the second dose, and to compare what cell populations are expanding out between these two different doses to understand if you give the patient the same CAR T therapy twice, will the cells look the same after each treatment? And surprisingly, we found the answer is no. So even if a patient's treated with the exact same product, we actually see a very different phenotype that can expand out at the second treatment. And this is very important to understand in terms of knowing that even if the first treatment fails, could it work with a second dose, and what are the signs we need to look for to know if a second dose could work? So all in all, we're just seeing these different dynamics between different products, and this is really just scratching the surface. There's many products for CAR T out there, and we're just looking 7 days after treatment, and we could be looking much longer follow-ups. But this is kind of this first glimpse of comparing these two products and looking at these important factors for response. So one of the things that's interesting about this is because looking at the infusion products themselves, because the markers that we see that are predictive of response are different for these two products, before we've actually treated the patients, it's certainly possible that you could think about generating both products, seeing how the T cells do with each construct, and making a decision later about which product to treat the patient with. Single cell, of course, itself is quite expensive, but as we learn from single cell, we could design more tailored assays to be able to identify the phenotypes that we've seen in our single cell data, to perhaps find cheaper ways to be able to quickly assess if we think a specific product's going to work for that given patient.